It is said that our bodies are made up of approximately 37 trillion cells. However, did you know that inside, there are a vast number of other inhabitants that are not our own cells? These microorganisms outnumber our own cells and are vital to our existence. The study of gut bacteria, or the microbiome, has recently attracted significant attention as a key to health and immunity. We are discovering that these inhabitants play a role far beyond simple digestion, influencing everything from mood to complex immune responses. In April 2026, a shocking piece of news broke that rocked the scientific community. It was the discovery that a unique ecosystem, created by specific bacteria, exists within the colon cancer tumor itself, functioning as a hidden biological driver. Today, we delve into the true nature of the bacterial fingerprint, imprinted deep within colon cancer, and explore how it might change our future. We are redefining the very essence of oncology and how we perceive the battle within. In April 2026, medical news worldwide reported the results of a large-scale comparative study that spanned multiple continents. This research investigated multiple types of cancer with unprecedented depth, involving a global consortium of oncology experts and microbiologists. The international research team utilized advanced multi-omic analysis to scrutinize the inner environments of various tumors, including lung, breast, and liver cancer. They were searching for hidden, shared characteristics that might exist across different types of malignancies. While the presence of bacteria was confirmed in many types of cancer, colorectal cancer stood out from the others in a statistically startling way. The data revealed a consistent, clear microbial community characteristic found in nearly every patient. This consistency across thousands of diverse samples from different regions was a major breakthrough. Researchers began to call this unique, recurring assembly of microorganisms the bacterial fingerprint of the tumor, a signature as unique as a person's DNA. The methodology was exceptionally rigorous. Scientists used 16S rRNA gene sequencing paired with high-resolution spatial transcriptomics to map exactly where these bacteria were located within the cancerous tissue and how they were behaving. They discovered that these bacteria were not merely surface contaminants or accidental inhabitants. They were deeply embedded within the tumor's core, existing in high-density clusters that appeared in virtually every colorectal specimen analyzed by the team. Why colorectal cancer specifically? The large intestine is naturally the most bacterial-rich organ in the human body. It hosts trillions of microorganisms that are essential for our survival, nutrient absorption, and systemic immune health. However, the tumor environment within the colon provides a unique, sheltered habitat. It seems to offer a comfortable hiding place where certain specific bacteria can flourish undisturbed by normal immune surveillance and protective barriers. These bacteria leave a biological imprint, a bioforensic signature that differentiates cancerous tissue from healthy cells. This discovery marks the end of an era in traditional oncological theory. We are moving away from viewing cancer solely as a cellular problem caused by internal genetic mutation. We must now consider the external microscopic inhabitants that drive its progression, survival, and even its resistance to treatment. The researchers describe the tumor as a hypoxic sanctuary. In these low oxygen zones, the normal aerobic bacteria of the gut struggle to survive, while specific anaerobic pathogens find an ideal and protected home to grow. In this sanctuary, the bacteria are protected from the body's primary inflammatory responses. They settle into the deep tissue layers, creating a stable and recurring pattern that we now recognize as the defining fingerprint of the disease. The 2026 study has fundamentally shifted the paradigm of modern medicine. We now understand that the tumor is not just a cluster of rogue cells, but a complex, thriving microbial ecosystem with its own forensic signature and survival strategy. By mapping these fingerprints, we are unlocking a new forensic archive of the human microbiome. This is the foundation upon which the next generation of cancer therapies and diagnostic tools will be built in the coming decade. One of the primary architects of this bacterial fingerprint is a bacterium known as Fusobacterium nucleatum. Historically, this organism was primarily linked to periodontal or gum disease, not considered a threat elsewhere. However, the 2026 research confirmed that this organism has a sinister talent. It can migrate through the bloodstream to the gut and bind directly to colorectal cancer cells, ignoring healthy tissue along the way. The mechanism is fascinatingly specific. F. nucleatum uses a unique protein called FAP2 to bind to a carbohydrate called galgalnac, which is overexpressed on the surface of colon cancer cells, acting as a lock and key. This binding allows the bacteria to effectively hitchhike on the cancer cells, following them as they grow and even as they metastasize to other organs like the liver, potentially carrying the fingerprint with them. Once inside the tumor, these bacteria don't just sit there as passive observers. They actively secrete metabolites and signaling proteins that promote cell proliferation. They essentially act as a fertilizer for the cancer's growth. By triggering specific inflammatory pathways, these bacterial inhabitants accelerate the tumor's growth and make it more aggressive, driving the progression of the disease at a microscopic level that was previously ignored. Perhaps more critically, these bacterial fingerprints may represent a sophisticated immune shield. They create a physical and chemical barrier that protects the tumor from the body's natural cellular defenses. By hiding behind this microbial matrix, cancer cells can evade detection by the body's T cells. The bacteria effectively provide a hypoxic sanctuary for the tumor to hide in plain sight, far from the reach of immunity. We've also seen that some bacteria within this fingerprint can break down drugs. They produce enzymes that render chemotherapy ineffective before it can even reach its intended target within the cell's nucleus. This helps explain why two patients with identical genetic mutations can have such vastly different responses to the same drug. The answer often lies hidden in their unique individual bacterial fingerprint. In other words, we must redefine colon cancer as an abnormal ecosystem. It is a lethal cooperation between our own biology and these external microscopic inhabitants, a partnership that science is only now beginning to map. A new page is being added to the definition of cancer, a disease caused by symbiosis with microorganisms. We are beginning to see the tumor as a living, breathing landscape of interaction rather than just a genetic error. This realization is rewriting the textbooks. We are no longer just fighting a rogue cell. We are trying to understand and disrupt an entire biological architecture that spans across different kingdoms of life. The bacterial fingerprint is the key that unlocks this new understanding. It provides the forensic evidence we need to build more effective, personalized treatment strategies for patients around the world. How do we isolate these fingerprints? In the bioforensic laboratory, scientists use isotopic labeling to track the metabolism of bacteria directly within the living tumor tissue, separating them from the host's activity. By extracting DNA directly from biopsy samples, researchers can use targeted metagenomics to sequence every microbial inhabitant, separating the signal of the fingerprint from the noise of the surrounding gut environment. Advanced fluorescent in situ hybridization, or FISH, allows us to see these bacteria in their three-dimensional context. We can observe how they form the physical architecture of the fingerprint within the tumor. This visual evidence is crucial for clinical acceptance. It proves that the bacterial fingerprint is a real, physical entity, a structural component of the tumor that we can now target with diagnostic and surgical precision. The most immediate application of this discovery is in the field of non-invasive diagnosis. If every tumor has a unique fingerprint, we can look for that fingerprint in blood or stool biopsies through liquid biopsy technology. Microbial biomarkers could offer a way to detect colorectal cancer at its earliest, most treatable stages with a simple lab test, potentially saving thousands of lives annually through widespread, easy-to-access screening. Beyond detection, the fingerprint can help predict how a patient will respond to immunotherapy. Oncologists can tailor treatments to the individual microbial ecosystem of each patient, ensuring a higher chance of success. By understanding the inhabitants of the tumor, we can bypass microbial interference, increasing the success rate of personalized medicine and reducing the toxic side effects of broad, non-targeted treatments. However, we must proceed with academic caution. We have not yet reached the stage where we can definitively say that removing bacteria will cure cancer. The biological reality is vastly more complex than simple elimination. Simply eliminating all bacteria with broad antibiotics could kill the good bacteria our bodies rely on for health. This could actually have a counterproductive effect on the patient's long-term health and treatment response. We are still in the critical stage of identifying the phenomenon. The next step is moving toward precise clinical application and the targeted disruption of only the specific, harmful inhabitants of the fingerprint. The goal is precision microbiome management. We want to enhance the treatment response by neutralizing the immune shield while leaving the rest of the body's delicate, essential inner world completely intact. The tiny fingerprints of bacteria etched within colon cancer tell a profound story about life itself. They reveal how complex our bodies really are and how closely we are intertwined with countless other forms of microscopic life. We are not isolated individuals, but vast, thriving landscapes of interconnected life. Science is showing us that cancer is not a simple enemy to be defeated, but a disruption of this intricate and delicate biological balance. Unraveling the mystery of the fingerprint is still in its early stages of discovery. But every step forward is a step toward transforming cancer into a manageable and symbiotic state of existence for the modern patient. Let us continue to listen attentively to the stories told by our inner ecosystem. The answers we seek are hidden within these microscopic fingerprints, waiting for us to fully understand and master them. Understanding the true nature of these tiny inhabitants may one day be the most crucial step in our collective fight against this disease. It is a journey of discovery toward a deeper, healthier symbiosis with the life within us. Thank you for joining us today as we explored the bacterial fingerprint lurking within colon cancer. Let us remain curious and vigilant about the intricate life that dwells within us. Goodbye for now.